friends, welcome back to my channel, and today in this video we are going to study about constructing a genetic map with 2-point test crosses. What do we mean by 2-point test cross? It is a test cross between two genes. 2-point test cross. That means a test cross between two genes. Now, genetic maps can be constructed by conducting such a series of test cross between pairs of genes and examining the recombination frequencies between them. How? Let's see. So here, some gene pair test crosses are given and their recombination frequencies are given. For example, the test cross between two genes A and B, the frequency is 50%, A and C 50%, A and D 50%, B and C 20, B and D 10, and C and D 28%. Now, in the last video, we learned that 50% recombination means those genes are either present on two separate chromosomes or at least in two linkage groups. If present on same chromosome also, they are present at two different linkage groups. So first of all, we have these data with us. So first of all, what we have to do, let's start with A and B. Because the recombination frequency is 50%, that means A and B are present in two different linkage groups. So first we need to plot that. In linkage group 1, A is present, linkage group 2, B is present. Now we have to see the gene pair A and C. Now here we can see that between A and C also, the recombination frequency is 50%. That means A and C are also present on different linkage group. What about B and C? Because we found that two linkage groups are there, A and B, till now. So we have to see that whether C is a separate linkage group or it is present with B, because it is not present with A. So if we see this B and C, so recombination frequency is 20%. So definitely C is also present along with B in the linkage group 2. And the distance is 20 map unit. Now come to A and D. With A and D, it is 50%. That means again, D is not present along with A in linkage group 1. What about B and D? So B and D is 10%. So that means D is also present in linkage group 2. Now the next question is where I have to map the D. I can map here also. I can map here also. That means I have two positions, either to the left of B or to the right of B. Now, what is the actual position of D? Is it to the right or to the left? For this, we need the distance between C and D. So here we can see the distance between C and D is 28%. So definitely, because it is 20 mu far, the distance between B and C. If I map D here, B and D distance is 10 mu. So that means C and D distance should also come 10 mu. But what is given here in the question? The distance is 28 mu. That means definitely D is not present to the right of B. So it has to be on the left of B. So this is the map. So A totally in a separate linkage group, and in the linkage group 2, we have B, D and C with their respective position. So this how we can use the recombination frequencies and the gene pair to map the position of all the genes. Now this one is homework for you. You have given multiple gene pairs with their recombination frequency and you have to map all of them. So one hint I am giving you, try to find different linkage groups first. Linkage group 1, 2. If it is more than 2 also, you have to first find the different linkage group. Let's start A and C. It is 50%. So definitely if I'm putting A in linkage group 1, I have to put C in linkage group 2. Then A and D, 14. So D is also present here. A and B, 10. So means B is also present along with A. A and E, 50. So E is not with A. Now we have to find C and E. So this is C and E, 8. So definitely E is present along with C. A and F, again 50. So that means F is not present along with A in linkage group 1. What about C and F? C and F is also 50. So definitely F is also not present in linkage group 2 along with C and E. So there is one more linkage group with F. If you want to confirm, because we saw like C and F in different linkage group, but along with C, E is also present. So definitely the recombination frequency between E and F should also be 50%. So here we can see E and F 50%. Understood? So like that, try to first segregate all the genes in different linkage groups, and then on the basis of recombination frequency percentage, try to map those genes. Do let me know in the comment section how you map the genes. So if you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.